All right, welcome back. We are going to be talking about chapter 2 in this section. Um, we're going to be talking about neural basis for cognition, and this is really going to be the only part of the quarter where we really address cognitive neuroscience. We're going to talk about implications or findings from it, but we're not going to really talk about the methodology or things as clearly after this chapter. If you've already had introductory psychology, some of this might be familiar, and it's always good to get another refresh anyway, but I wouldn't begrudge you if you feel like you understand it well enough that you sort of skip over some of these. All right, so um, I'm going to cover it across these recordings, these lectures, um, the sort of the main impetus for really starting to study the brain the way that we do today. I'm going to describe how the brain cells, the neurons, work. The main structures of the brain will come next, and then we'll talk about all the things that the cerebral cortex does, and then we will talk about some of the sources of evidence about the brain, how we know what we claim to know. All right, so let's jump in and talk about why we study the brain. There are a lot of reasons why we study the brain, and people have been talking about at least the outputs of the brain probably since, humans became human. Uh, but really, the seminal thing that happened that really started turning doctors and other people toward the importance of understanding the structures of the brain was the case of Phineas Gage. Some of you may have already heard of Phineas Gage, but for those who haven't, he in the early 1800s he was working on the railroad, and he was the supervisor of a bunch of employees who were trying to blast out some, New England granite to make the roadbed. And to blast the granite, they would take blasting powder and they would put it into drilled holes, and then they would use that metal tamping rod that you see Phineas holding there in that picture on the right, and they would tamp it down. They would throw that rod down into the hole as hard as they could to really pack that powder down, so that then when they ignited it, it would blow up the rock. Well, two workers were tamping the powder down, and Phineas was overseeing it, and the blasting powder ignited, and the tamping rod was like a projectile, like as fast probably as a bullet can travel. It went through his cheekbone on his left side, as depicted in this right picture, and the picture in the middle, I mean. So it went under, went through his cheekbone on his left side, through his eye socket, and that's why he has no eye in the picture on the right. And then it came out the top of his head, and you can see his skull on the left, where there had been a really big hole blown out. A lot of bone had grown in to fill in that hole by the time he died. So that shows that he lived a long time after this injury. Our imagination tells us that he wouldn't have lived through an injury like this, but apparently the tamping rod moved really quickly and it didn't cause an infection that he died from. So he made it through this injury, and then people observed that his personality had changed, that he didn't have as good of control over his emotions as he used to have. Now, looking back in retrospect, people think that the tamping rod probably severed some of the nerves that connect his limbic system, where emotions come from, to his frontal cortex and the part of his brain that decides on the wisdom of expressing what the limbic system might be feeling. And so, with those nerves severed, he expressed his emotions a lot more. He had some motor problems. He had to walk with a cane and some other effects afterwards. But basically, he, really recovered well given the injury that he had sustained. Having these changes in his behavior and him living through the accidents, the researchers could see that there were effects, long-lasting effects, effects that permanently were in place now, really spurred the idea that there are probably particular structures of the brain that you can live without maybe, but it will have an impact on your behavior. It'll have an impact on the way that you think about things. So he was really a big impetus for the beginning of you know research into the brain and the structures of the brain and how exactly the brain functions. So let's start with the smallest component really that can contribute to behavior, and that would be neurons. So here we have a picture on the right side of the screen, a cartoon drawing of a neuron, and I've got my little labels and an arrow so we can follow the progression of information from the dendrites, which are at the very top of that diagram. And they are called dendrites because that is Latin for roots. And you can see why they would say, hey, these look like roots, wouldn't, can't you? The dendrites look like roots. They're also like roots in the sense that they collect information and bring it into the cell body. So the dendrites are responsible for collecting information, collecting incoming signals from neighboring cells or from receptor sites, other receptor sites. So that information is conducted from the dendrites into the cell body, which is that round part with the nucleus in the middle. The nucleus is like any other cell in your body. It has your DNA and your RNA and all the mitochondria and all the things that need to be in any kind of cell, right? But the thing that this cell does differently is that it's a decision maker. As information comes in from the dendrites, the cell body has to decide whether that incoming information is sufficient to warrant passing that information along to the neighboring cell. If it does decide that it's worth passing along, it's going to send that message down the axon, which in this picture is encased with the myelin sheath, those blue bubbles. The axon's in the middle of it. So the information will go down the axon and then ultimately end up in those axon terminals that you can see at the very bottom of the picture. So all of the information is progressing from top to bottom in this diagram. The dendrites collect the information, the cell body makes a decision about it, and then the response is conducted down that axon and out those terminal fibers. So you have long chains of neurons in your brain, right? You have lots of neurons connected to each other, but they aren't actually touching each other. If they were actually physically touching each other, they'd be all one structure. So there's little microscopic spaces between the axon terminals of the sending cell and then the dendrite cell walls of the receiving cell. So you can see a chain of two neurons in the top part of this picture, and then if you look at the bottom part of the picture, you can see a zoom in on one of the axon terminals, and then you see this synaptic gap between that axon terminal and then the dendrite. Now, electricity does not jump gaps. And so everything in the cell body, everything in the neuron has been electric. From the moment it's collecting information at the dendrite, through the processing in the cell body, and then the conduction of an action potential down the axon, all of that was electric. But when the electricity gets down to the axon terminals, that electricity causes those synaptic vesicles that you can see in that zoom in picture to migrate to the cell membrane there at the bottom and then rupture and release the contents of the synaptic vesicle out into the synaptic gap. The contents of the synaptic vesicle would be the neurotransmitters. These are molecules that are specifically designed to lodge into certain receptor sites. So a axon that's sending out a particular neurotransmitter can only communicate with dendrites that are capable of receiving that neurotransmitter. This is a really elegant way to ensure that you don't just accidentally activate the whole brain at one time, right? If you're trying to have a particular thought about a particular subject, you'd like to, as much as possible, keep the relevant neurons talking about that thing inside your head rather than passing it on to everyone. So this is a really elegant way to make sure that that happens. Now, the way that information gets passed is that these neurotransmitters change the postsynaptic membrane. Those neurotransmitters lodge in those receptor sites and convince that dendrite that something important has just happened. Now, the change in the postsynaptic membrane may be to become excited. It could be that the neurotransmitter that has been secreted is something like acetylcholine, which is a neurotransmitter that is really important for the formation of new memories and for learning. And so if the sending cell releases acetylcholine and the receptor site receives it, that is going to trigger that receiving cell to become excited and pass the information along, share that with the neighboring cell. Other neurotransmitters are inhibiting. So certain members of the endorphin class are inhibiting, things that are the type that are activated by opioids, right? That are painkillers. If you have an injury, your brain might secrete endorphins out of the sending cell, and when they lodge into the receiving cell is inhibited from sharing that information. And that's how endorphins reduce pain. They actually cause the brain to stop passing the message that pain was experienced. So the neurotransmitters can change the postsynaptic membrane by exciting it or by inhibiting it. So here are some phrases to know. If there's sufficient ionic flow, now I am not going to bog us down with a really great discussion about ionic gates and potassium channels and a bunch of other things that I'm a cognitive psychologist, not a cognitive neuropsychologist. So I'm just giving you the bare minimums that you need to know to understand how the neuron works. If you're really interested in this stuff, this is in neuropsych. So you can take that class and you can learn more details. But so the ion gates open and close and provide the proper context for an electrical impulse to happen if the input coming into the dendrites is sufficient to surpass that cell's threshold. So every cell has a resting state where it's sort of like in normalcy. If incoming information exceeds that normalcy, that will trigger that cell to pass information along. So what is currently the resting state might be different than what other resting states are. For example, while you're sitting in a quiet room listening to music out of your speakers at a reasonable rate, your brain is accustomed to that sound and it becomes the resting state. It would need more stimulation in order to trigger a response than what it is currently experiencing. It becomes accustomed to it. Weirdly, if you go someplace really loud, you can experience the same thing. You could be in a bar where the music's really super loud, or I don't want to say concert because that oftentimes is like really loud, but something more like a bar where people are talking, a lot of things are going on, and you become so accustomed to it that it takes a lot of excess noise to trigger your brain that this is above threshold. I need to respond to this noise. I've gotten so used to tuning out all this extra noise, and so that resting threshold can change depending on what's going on around the brain at the time, right? If you're in a quiet situation, it'll take less to exceed the threshold. If you're in a noisier situation, it'll take more. If you're already in a room with a lot of bright light, it's going to take a lot more light to activate you and make you realize, hey, I'm seeing something different. So that's what I mean by that resting state can change. Once it surpasses the cell, whatever its current threshold is, that'll cause the cell to have an action potential. Now, action potentials are an all or nothing thing. You either have an action potential or you don't. There's no such thing as like a partial action potential where you sort of respond because this is a sort of mild stimulus. There's no such thing as that. So you either fire or you don't, and you have an action potential or you don't. As a result of that, you have intensity needing to be represented in some fashion, right? So if a stimulus is more intense, then more cells will be firing, and they will fire faster than when the stimulus is more mild. So that's how we represent intensity. It's not by the individual cell firing harder or something. It's about how many cells are firing together and how quickly they're firing. All right, so if we have a really intense stimulus, all the cells will be firing that are relevant to that as quickly as they can. One of the implications of that is that you can reach a level of incoming stimulation where you can't represent that full intensity anymore. If you've ever heard of somebody who burned out their retina because they were staring at the sun, and you thought to yourself, didn't you realize that was going to hurt your eyes? Like, why didn't you move your eyes? Every cell it was probably firing too bright, look away, as fast as they possibly could, as fast as like too bright, too bright, too bright, fast as it possibly could. But there was still more intensity that could have been represented, but the brain was limited by this refractory period that every cell has, and there's only so quickly that they can respond. So there are certain kinds of really intense stimuli that we probably don't really fully detect because of that refractory period that we have. Um, let's see, my thing isn't forwarding. All right. So the strengths of the synaptic connections are adjustable. So how well a receiving cell responds to that sending cell's message can vary, right? Depending on things like experiences that you've already had, right? So if you're using a part of your brain a lot, let's say that you're learning a new language, and so you're using parts of your brain to memorize the vocabulary and understand the grammar and all those things that go along with learning a new language. Using it a lot will make those connections a lot stronger. Now, let's say you don't really use that second language for the rest of your life. um, 20, 30 years have passed. The strength of those connections will weaken, right? So you would have to sort of reteach yourself those associations. And the good news is you can reteach yourself. It's a process we're going to talk about called rapid relearning, where you might think you forgot something, but you learn it a lot faster the second time, because what you're doing the second time is just reminding your existing synapses, hey, you already know this. Re-establish the strength of that connection that you once had, right? So part of the strength of our connections are due to being in a moment where you're using that a lot, and so it really strengthens those connections, or being in a period of time when you're not really using it, it can weaken those connections. Usually we don't think they go away altogether. So you may think you have fully forgotten something, but more than likely you can rapidly relearn it if there is a synapse dedicated to that piece of information. And that's where synapses are crucial for learning. Every single thing that you know is represented by connections between neurons. And so luckily these things are really flexible and are very responsive to practice and experience. There are more than 100 neurotransmitters that have been identified so far. Probably about 10 of them are really super relevant for psychology, and probably like 3 or 4 of them are really important for like cognitive psychology in particular. But every day they're finding new things that are relevant to neurotransmitters, finding out which ones have an influence on this behavior, or which ones are influenced by this dietary intake, or different kinds of things. And so it's a really growing field, trying to understand what our brain chemicals really do to our mood, our learning, our motivations, things like that. All right, well, that's a good little overview of neurons and how they exactly work, with some vagueness implied. The next segment, we're going to talk about the main structures of the brain. We'll start off with just the big, gross ones, and then we'll start getting into more detail. So I'll see you then.